I'm going to talk about the U.S. regulation and safety assessment of food additives with the consideration of genetic toxicity recommendations. I think everyone here in the audience today, and I think those who are online currently, especially those that are attending from Canada and U.S. where it's still very early, so thank you. And this is my disclaimer. The findings and conclusions in this presentation have not been formally disseminated by the FDA and should not be construed to represent any agency, determination, or policy. The mention of commercial products, their sources, or their use in connection with material reported herein is not to be construed as either an actual or implied endorsement of such products by the HHS. Since we have different sectors here in the audience and online today, I did want to speak briefly about just some basic definitions of food additives, food contact substances, and impurities, because I think it's important to state these as we will discuss when we go further in the workshops this afternoon and the following 2 days. So FDA, when they defined a food additive, it doesn't matter how the substance got into the food, whether it was direct or indirect. It's the fact that it got into the food that we care about. Your direct food additives are those that are on your labeling, and the indirect food additives, which is what I do day to day, those are the ones that is the result of food contact use, and it's not intended to have a technical effect on the food. These indirect food additives are also known as food contact substances. These are discrete substances. They're components of the food materials. So when I regulate by specific substances, not the entire material. And this is by intended use. And some examples would be monomers, catalysts, et cetera. Impurities or constituents are actually those of the impurities of the food contact substance, and they're the result of breakdown products. So this distinction is very important when we talk about the Delaney Clause. Delaney rules my day each day as I provide risk assessments, and it was enacted in 1958. It prohibits the FDA from approving any additives, direct or indirect, that induce cancer in humans or animals after oral ingestion. So that distinction is very important when you talk about those who do food safety at the FDA. So although the Delaney Clause results in a zero risk standard for food and color additives that have been shown to be carcinogenic, FDA does apply a de minimis cancer risk standard to those constituents and impurities. The FDA's constituents policy allows for cancer risk assessments to be conducted under the general safety clause for a constituent that has carcinogenic potential without triggering the Delaney Clause. We use the safety assessment framework that was derived, we affectionately call the Red Book in 1983. I was 2 years old when it came out, just to give it hasn't been around that long. We start with a pre-assessment, so we identify chemicals first. When I do my risk assessments, I always know the exact identities of these chemicals. Then I perform my hazard identification, so we're screening for the substances of toxicological concern. I have a chemist at the same time performing an exposure assessment, so I know estimated exposure levels using either actual migration studies or assuming 100% migration. And then if needed, depending on those exposure levels, we may then go into a dose response assessment and determine that threshold value, which over CIFSAN we call an ADI value. And then we'll do a risk characterization, so assess that margin of safety if need be. Throughout this entire framework, we're using expert judgment, the totality of the data and information available, a weight of evidence approach, and we consider this framework sufficiently conservative. When we start our safety assessments, we spend we have a 120-day review. That first 30 days, we spend a lot of time considering what the chemical structure and toxicokinetic profile is. So what is that chemical structure? What is the class of the agent? What are some of the chemical features, solubility and stability? What's the expected metabolism, reactivity, and biological activity? And then if you're thinking about nanoparticles, there are special considerations for their physiochemical properties and need for particle characterization. And then consideration of the bioavailability and target tissue of the substance after oral exposure. This table is our tiered exposure-based safety assessment approach. It's online. It's guidance for industry that we use. On the right side, you'll see our recommended, not required, recommended toxicological testing. It shows the tox endpoint that we're interested at that level of exposure, and then the recommended assays. On the left-hand side, you'll see the dietary concentrations, and it goes through tier 1, 2, 3, 4. For those substances that below 0.5 parts per billion, we had a publication by Cheeseman et al. In 1999 that we still use today that said FDA determined that 0.5 parts per billion was an acceptable threshold for an unstudied compound. But at that first level, we're still asking our notifiers and our reviewers, even though testing may not be recommended, we're still looking for a literature search, and with that point, a toxicological endpoint of being carcinogenicity. At tier 2, that's the 0.5 to 50 parts per billion dietary concentration. We're starting to request those in vitro genetic toxicity studies. We prefer to see an Ames test and then either a Chromab assay that's in vitro or an in vitro mouse lymphoma TK assay. When we start moving into the 50 parts per billion to 1 parts per million range, each of these recommendations are required on exposure level. So then we're starting to ask for an in vivo test for Chromabs, and then 2 subchronic oral toxicity studies, one being in rodent species and one in a non-rodent species. If you don't have both species, obviously that would increase your safety factor. And then anything above 1 part per million, additional testing could be required or determined by a case-by-case basis. So there is also a cutoff level of concern that we work with. The FDA's Genetic Toxicity Assessment Committee concluded in 1999 that in general, a substance whose use results in an estimated dietary exposure of less than 50 parts per trillion would generally be expected to not pose a safety concern, even if the substance is found to be genetically active. And so the scientific reasoning for this is that 85% of all carcinogens with alerting structures have a median toxic dose corresponding to less than 10 to the negative 6 risk of 0.05 parts per billion in the diet, or 50 parts per trillion. And this cutoff is not considered an absolute cutoff, though. It's applied on a case-by-case basis and evaluating the totality of toxicological data that might be available. Regarding our constituents or our impurities of the food contact substances, this is when we start getting into our cancer risk assessment. If you have a cancer study and it's either positive or equivocal, then we derive a lifetime cancer risk for that chemical. And that kind of gets into Stephanie's discussion of some of the studies that she does at NTP. The LCR is an upper bound lifetime cancer risk to humans from an exposure to a constituent. When calculating the LCR, we use tumor data from the most sensitive species, strains, sex, and study. We assume tumors arising at multiple sites are independent of each other. And we calculate a unit cancer risk, a UCR, which we can provide to notifiers that ask. And this is the slope of a straight line drawn from the lowest apparent effect dose to 0. And then we calculate the LCR based on that estimated exposure, whether you have an ADI or what we call as a CEDI, so that's your cumulative exposure of that substance from all intended uses of that. An LCR below or within the 10 to the negative 8th level or 10 to the negative 6 is considered a historically acceptable cancer risk level for a carcinogenic impurity, and that's either with incremental or cumulative exposures, respectively. I have just enough time to give 2 case examples. The point of the case examples aren't to get into the nitty-gritty risk assessment of these 2 examples. It's more to just give an example of how we look at genotoxicity and carcinogenicity studies. The first one is aspartame, which Diane kindly was the chairperson for JECFA, and actually my new supervisor, Shruti Kabadi. So thank you both for all the time you spent on that and the conclusions you came out with in July. It is one of the most studied food additives in the human food supply. The work they did for us was large, so thank you. It is a direct additive, so not something that I review day to day since I do indirects, but it's a sweetener. It's authorized for food additive use in the US under certain conditions of use. It's your NutraSweet or your equal. So following oral exposure, aspartame was found to fully hydrolyze in the GI tract of humans and animals into 3 metabolites, and these metabolites actually form at levels much lower than those derived from common foods. There was a mixed result of genetic toxicity. However, there are major limitations with study design, and no systemic exposure to aspartame is expected upon oral exposure, so we had no concern for genetic toxicity from aspartame after oral exposure. There was a conclusion of no concern for carcinogenicity for animals. There was 12 CARC studies. All of them had deficiencies. The issue et al. 1981 study in rats was the closest to meeting current testing guidelines and showed negative results. So there was unconvincing evidence of association of aspartame exposure and cancer and non-cancer endpoints in humans. And so the conclusion for JECFA with the new evaluation was that it didn't actually change the ADIs or raise any other new safety concerns. My second case example is acrylamide, which actually Franz discussed as one of his examples, so it must be a popular one today. It's a monomer or constituent of polymeric food contact substances intended for certain uses. It has an estimated mean dietary intake from foods of 0.36 micrograms per kilogram per body weight per day. As Franz mentioned, it's generated in a wide variety of foods, breakfast foods, potato chips, coffee, pretty much everything that we love to eat. It did show positive genetic toxicity data, and it was reported carcinogenic in a 2-year rat drinking water bioassay. And so because of this, we derived a UCR value for this substance at 0.72 milligrams per kilogram per body weight per day. There was a basis of no safety concern for exposure to acrylamide from our current food contact use, and that includes an EDI of acrylamide from food contact use being much less than the current estimated dietary intake just from regular food consumption. The LCR for the EDI of acrylamide is less than the agency's historical acceptable risk level of 10 to the negative 8 for incremental exposure to a potential carcinogenic impurity, and we can still take substitutional exposures for acrylamide. I did want to mention that in CIFSAN, we do not consider risk-benefit analyses when we are doing food safety. So CIFSAN spends a lot of time and resources in exploring new approach methodologies for evaluating genotoxic potential. We have a NAMS working group that's led by Susie Fitzpatrick. And as we've discussed today, NAMS can include those in vitro and chemical or in silico methods, and some examples are shown here on the right. QSAR and read-across are useful to support safety assessment in a weight of evidence approach. I think currently, every review I've been doing, I've been using predictive modeling when I begin the review now. FDA does promote the development of alternative test methods to support replacement and reduction and our refinement of animal testing. And as my mentor, Dan Levy, said, we should remember that in vitro genetic toxicity testing was our first NAMS that the field brought up. So I thank you for your kind attention. I thank those who helped me prepare these 10 slides since we only had 10 minutes. That was Jason Ongst, Paul Hungenfort, and Shruti Kabadi. And I also had nice discussions before this with Christy Mullendon-Jacobs, Dan Levy, David Kirkland, and then also Stephanie Smith-Rowe. So thank you for your kind attention. If you have any questions, I put my email at the bottom. I'm happy to chat. And I think we're ready for our panel, right?